Okay, guys, we are going to do a quick recap and overview of muscle excitation, contraction, relaxation. I'm going to try to tie these pieces together for you in a very coherent picture, and I hope this makes sense. I'm trying a very new approach to this, using just the pad to draw. I've created some pictures, pre-existing pictures that I drew before the lecture, just to kind of show you how all these things kind of come together. So I hope that this is a success, and I hope that this shows you how all of these things interact, and we get a better understanding of contraction, and you guys will be able to replicate this on the exam perfectly. So let's start with this guy here. We're going to look at the somatic neuron. Obviously, we know that when a neuron receives signal, we're going to start getting some signal coming from other neurons, telling the neuron to fire an action potential, or potentially telling the neuron not to do anything. And what's going to happen is these signals are all going to come down, come down, come down, and at the axon hillock here, the trigger point that we've talked about, I'm just going to put AH for axon hillock. That's going to process this information, and then essentially it's going to fire an action potential, and you guys know what that is because we've talked about the action potential and how it looks time and time again in class. So we're going to have that figure again, and action potential is going to be fired. It's going to travel down the length of the axon until it gets to this portion here, which is called the axon terminal. Okay, and that's these finger-like projections right here. So when we get to the axon terminal, we also are met with the muscle fiber. So this part of the structure that we're talking about here and here, okay, encompassing both of those, is the motor unit, and this is where we have the muscle fiber that meets the neuron. Now, if we kind of zoom out a little bit, let me bring this center again. I wanted to show you how the motor unit interacts with the other parts of the muscle. So on the left-hand side here, I'll draw a little arrow, right? We kind of zoom out a little bit so we can understand where the motor unit rests in this giant mess of muscle, right? Because muscle is very complex. There's lots of layers, and a lot of people kind of get confused on it. So I drew a bone, and then here I'm going to highlight this. Here we can see I have the muscle. Okay, and if we take a cross section of that muscle, so let's just say I cut a line right through the belly of that muscle, and we open it up, we can see that inside that muscle there's these other anatomical structures, and we call those fascicles. Okay, so now if we took one of these fascicles, and let me just highlight this here. Okay, we took one of these fascicles and we pulled it out like it was your sock drawer. All right, we pulled out that fascicle, we would see that that fascicle has other anatomical pieces in it, which you can see right here. Okay, and those are the muscle fiber. So it's important to know that the fascicle, I'm going to highlight it, the fascicle, it contains the arteries, right? It contains neurons, and it contains veins, so that's like a big portion of the blood supply, and the removal of blood and metabolites and things like that. Inside the fascicle, we also have another subunit called muscle fibers, right? Or there's another name for it that's pretty popular, right? And that's the muscle cell, right? So, and inside that muscle cell, we have, I'm going to kind of go over here to the far right. Inside that muscle cell, we have additional, highlighted in blue, I hope you guys see that right there. We have additional subunits, which are the contractile elements, all right? That's where your myosin and your actin and your sarcomeres are located. Okay, so I just wanted to kind of draw this picture here just to kind of show how everything is associated, right? Like the the big part of the muscle, the fascicle, the muscle fiber, and then the myofibril, which is, let me change the color here. So the myofibrils are in this area here, right? So they're inside of the muscle fiber, right? So I'm just going to put a big green highlight there to make it really pop out. All right. So now let's kind of zoom out in a little bit. Okay. Now we're going to focus on the axon terminal. Okay. So this is representing the bottom of the neuron. Okay. So when the action potential occurs, okay, we just have red lightning. That's the universal symbol for action potential, just in case you were wondering. It travels down the axon to the terminal, and our point of interest is going to be these axon bulbs, right? So we're going to look at kind of like the physiology that occurs there. So what I'm going to do now is I'm going to draw this box right here. And what that box means is now we're going to take our microscope and we're going to zoom in even more. Okay, so not only are we talking about the anatomy of all of this, but we got to talk about the physiology as well. So if we zoom in, just trying to find my notes here to make sure that they're aligning with, because we had two different lecturers on this, so I just want to make sure everything's aligning. If we zoom in on this axon terminal and we look at one bulb, as I indicated here, right, let me just draw it one more time so everyone's following along. We see that inside of this bulb, let's zoom out a little bit, we have these vesicles. Okay, and what's inside of these vesicles? I know right now you're saying, I know what's inside of those vesicles, and if I were in class I could answer it. So I'm going to draw these things in here. Inside of these vesicles we have what we call neurotransmitters, right? And look at those, are perfect circles. That's pretty cool. And there is a specific type of neurotransmitter that is in these vesicles, and we're going to call that what? Draw a line. Did you answer it? Yep. It's going to be acetyl. Got to draw it a little thicker. So, choline. I think there's an E at the end. So inside of these vesicles, we have acetylcholine. So again, action potential comes down, as indicated by this red line. Okay. Red line, action potential. That comes all the way down into the axon terminal. If we zoom into this bulb here, we see that we have these vesicles containing acetylcholine, and they're waiting for that signal to come down. Okay, so let's just kind of keep our place there, and let's go back to the motor unit. All right, so again, we have the axon bulb. Okay, we have the vesicles. Inside the vesicles, we have the acetylcholine, the neurotransmitter. Okay, let me just tap on these a little more. Tap, 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 tap. Okay, so now we get to the other really, really important structure. All right. And we got to talk about some of the physiology here and what is occurring here, so that we understand how this neurological signal is turned into like a chemical signal. Okay, so if we look at, let me zoom out so you can see what we're looking at. I drew the muscle fiber. So you can see that the muscle fiber has, let me highlight that for you guys. Let me change the color because that's just really a hideous color. The muscle fiber has the sarcolemma, right? So that's a membrane. Here I identified the muscle fiber. You can see that, skeletal muscle is multinucleated, so you can see that there's nuclei kind of all over the skeletal muscle. And then when we get to this kind of open-ended area here, this is the myofibril area where the contractile properties are. Okay, so we know that there's myosin, actin, and the sarcomeres that are located in these sock drawers, right? So we're just going to keep referring to these things as sock drawers, just things that you can keep pulling out. So that kind of orients you to the picture. And now we look at the axon bulb. So in green here, we can see that on the other side of the synaptic cleft, we have these receptors that are designed specifically to receive message from acetylcholine. And next to those receptors, we have these ion channels. I'm just going to put ion C because I'm going to run out of space here. Ion C. Okay, period. Now, just like our other lectures, you can see that they have cute little hinges and cute little doors. Okay, so here's the hinge, here's the door. And we know that prior to the neurotransmitters being released, these ion channels are closed. Okay, it's also important to know that within this synaptic cleft here, we have very high levels of sodium. Okay, I'm going to put sodium. Okay, and on the inside of the cell, we have very high levels of potassium. Okay, and this creates a chemical gradient, and it, obviously there is a difference in charge on the membrane as well. So, these being ion channels, we understand that when they open up, when there is a signal that tells them to open, the ion door is going to, let's find this guy right here, he's going to swing open, okay, and that's going to let sodium then flood the cell, okay, but I don't want to get ahead of myself, so let's just kind of look a little more at structural components here, okay, so we have the bulb, we have the vesicles, we have the acetylcholine inside, we have the receptors as indicated here. We have the ion channels as indicated here. We know that there is sodium floating around. We know that there's potassium on the inside of the cell. And now let's zoom out a minute here, and we're going to go over here. So now when we get into the myofibrils, or the filaments, the actin and myosin contractile properties, we're going to pull out one more drawer, okay? So let's imagine that we pull out this sock drawer now. Okay, and we'll draw that there. Now, this is where we find what we talked about in class, which are those Z lines. Okay, and we know from Z line to Z line is a sarcomere. Okay, so I'm going to put Z line and Z line. Okay, and this together equals a sarcomere. This is hard to write on this. Okay, and if we zoom in, we know that coming from the Z line are our thin filaments that we call actin, right? And they go in both directions of the Z line. And then we have, in the middle of that, we have our thick filaments. I'm just going to draw it like this, okay? And we know that those thick filaments are the myosins, okay? So within this structural component of the muscle, we have the contractile property, right? The filaments. And the filaments are a part of the myofibril. Okay, does that make sense? So something else that we have in here, let's pull out one more drawer. Okay, and the other thing that we have that kind of overlaps this structural component, being the filaments, right? We have another layer, like a superficial layer, that covers the contractile properties, all right, the filaments, and that would be something called the T-tubules, all right? So I want you to imagine that this is kind of overlaying the myosin and actin, okay? It's like resting over it. And then on both sides of the T-tubules, we have these structures called the Sarcoplasmic Reticulum, okay? And then we have a T-tubule here, and this also has a Sarcoplasmic Reticulum on both sides of it, okay? And then basically you would see underneath those T-tubules, you could see like the myosin right right? Myosin. I'm sorry, those would be actin. And then let's go a little thinner on this. And then myosin structures. Okay, underneath that. So you can see here that we have the T tubule, okay? And then here we have the sarcoplasmic reticulum, and we know that in the sarcoplasmic reticulum is lots of sodium, okay? And we also know that when you have a sarcoplasmic reticulum, so I'm going to put SR, and then a T tubule, I'll put TT, let's zoom in, this is what's so cool about this, and another sarcoplasmic reticulum, okay, we know that these three things together are called the triad. Okay, so let's zoom back out. Okay, so now let's kind of go over to the T tubule and look at the T tubule structure. So now we're going to kind of zoom in on this section right here. Okay, are you guys following me? So we're going to zoom in on the sarcoplasmic reticulum and then a T tubule. So I'm going to move over to the right hand side here. Okay. So I've already labeled this. So here we can see, let me make sure this is thin enough. The sarcolemma. Okay. And then here we can see the T tubule descending down into the belly of the muscle. So T tubule is what we call those are, invaginations of the skeletal muscle, and we can see that it is moving down into the muscle. So its structural its structure is functional because it's going to allow the action potential and the depolarization that occurs over here, right, when we have the acetylcholine being released and it binds to these receptors. We're going to have an action potential fired. That action potential is going to basically propagate all the way down, right, down the sarcolemma, and then it's going to go down into the T tubules. Okay. Now here we have, zoom out. Here we have the sarcoplasmic reticulum. Okay, that's kind of this area here. I'm going to fill it in with these lines. And then it's this area here. Okay. And the sarcoplasmic reticulum is on both sides. You see that? Of the T tubules. Okay. And inside here, we have lots of calcium. Okay. And we have calcium ion channels, and you can see that. Let me zoom in here. We can see that here. And these ion channels, they have hinges and they have doors, and they have hinges and they have doors, and they have hinges and they have doors, and they have hinges and they have doors. And calcium cannot get out, and it cannot flood the cell until it receives a signal that allows these doors to open. And if you're thinking, well, yeah, it's going to receive the signal from the neuron that is firing an electrical signal, and that's going to be converted into a chemical signal. Okay, so we have an electrical, let's look back over here. We have an electrical signal coming down. Okay, that's going to activate these vesicles. Those vesicles are then going to, let's zoom in, release all this acetylcholine. Okay, it's going to cross the synaptic cleft. Okay, and it's going to bind to these receptors. Now, once the receptors bind to the ligand, which is the neurotransmitter, that's going to cause these ion channels right here to open their doors. So this is the door opening. Oop. Okay, the door's going to open. Door swings open. Okay, door swings open. So here, we'll do this. Now that it's swung open, we'll erase this door here. Okay, so that door's gone. That's gone. That's gone. That's gone. Okay, the doors have swung open. Let me draw this again. The doors are open. And now what happens is all this sodium that is floating around out here, let me fix this, gets to flood inside the cell. Okay, so the sodium will go inside the cell. The sodium will go inside the cell. Okay, the sodium will rush inside of the cell. Okay, so again, just to review, the electric signal comes down from the neuron. It stimulates the vesicles. The vesicles release acetylcholine. Once the neurotransmitter binds to the receptor, it opens the ion channels. And once the ion channels open up, the sodium can follow its concentration gradient into the cell. And what happens then, just like what we saw with the neuron, I'm going to draw it right here. Okay. We have depolarization of the sarcolemma, and that depolarization, I'm right over here now, is going to follow all across, hit something wrong there, all across the sarcolemma. Okay, it's going to spread out, and it's actually going to go in both directions. Okay, so it's going to happen this way and this way. All right. So now, when that message spreads out and it basically travels the whole course of the sarcolemma, then it's going to reach these T-tubule structures here. Okay, and when that message is coming down through the sarcolemma, it's then going to travel into the invaginations, which is the T-tubules. Okay, now that action potential coming down is what's going to cause these doors to flip open. Okay, so these doors were closed and now they flip open because the action potential was carried down the sarcolemma to the T-tubules. The doors are now open. The doors are now open. All that calcium, I'm going to erase the existing doors here so that it doesn't confuse you. Okay, all that calcium that is now in the sarcoplasmic reticulum is going to be allowed to leave the sarcoplasmic reticulum and flood the cell. All right. So we're going to go to one more component and then stop there. And I don't want to oversaturate you with all this information, but I'm just trying to draw you pictures that kind of put all this together. So now what happens is the calcium that is in the sarcoplasmic reticulum can leave. Okay? And that calcium can now interact with the sliding filaments, which are actin. You guys should recognize this. Actin and myosin. Okay, so now that calcium is allowed to leave the cell, or leave the sarcoplasmic reticulum, it can now interact with the regulators that are responsible for controlling contraction. And that's what we'll get into on the next lecture. We'll start talking about those regulators and how calcium interacts with them. So I'm going to stop here. That's a lot of information. I just kind of want you to understand the whole picture. Okay, so I'm trying to show you guys, you know, from a microscopic level to more of a kind of a larger gross anatomy level, how the neuron and the motor unit interact. I want to show you, I wanted to show you how important it was to understand the physiology that occurs on the axon terminal and with the axon bulb, how this part in particular creates the action potential and the depolarization of the muscle membrane, which is the sarcolemma, and how that travels down into the T-tubules, thereby releasing calcium from the sarcoplasmic reticulum. Okay, and then we'll stop there. So I hope this helps. I hope it doesn't confuse you more. This is the first time I've done this, so I hope it's good. Have a wonderful day.